Good morning, everyone. I'm Tom Carmichael. I'm Chair of Neurology, and this is just a quick intro because we have so much great stuff coming down the road. So it's a pleasure to welcome everyone to the 2025 Turken Research Awards and Symposium. This is Ida and Sam Turken Foundation, mediated through Beth DeVermont. And for over 3 decades, it's supported a really strong focus on up-and-comers, new science, and an integrative approach. And I'll note that in a minute, an integrative approach across the Alzheimer's and related dementias field. And I'd like to recognize two people from Alzheimer's LA that Jason Hinman will further mention later. Steve Klappholz, who's joining us, and Jennifer Schlesinger, who's parking. So it'll be great when they come in. This is really a pivotal moment in Alzheimer's disease, and it's exciting to have this symposium at a moment like this. Most of us know that there's the two and first ever disease modifying therapies that have come through. And we are up to our eyeballs in a really comprehensive care program at UCLA that Jason and I helped design for the whole UC system, really, for this care. And we know that they're unfortunately only very modestly effective, but they've seemed to have swung open a door at big pharma and biotech and a renewed interest in understanding not just disease-modifying therapies, but the subtleties of inflammation that the only modest effect of these therapies have revealed as something we need to understand more. And we'll hear in this today set of studies the whole spectrum of what's probably multiple disease entities in Alzheimer's disease: proteopathies, inflammation, synaptogenesis, and neuritic dysfunction, oxidative stress. And we'll hear about all these in some of the posters today. So just in conclusion, this is sort of a collective effort. It's a spanning vision we have to understand Alzheimer's disease, to develop new therapies, to understand progression, the genetics, the environmental interactions with genes that hasten or retard that. And it's exciting to be a part of that with the youth, the mid-level, and the senior people present. And so welcome. We'll have a great morning. It's going to be at this new venue, so it's all close, and I look forward to the interactions that we'll have. So thank you, and Dr. Hinman.
Thank you, Dr. Carmichael. And welcome to everyone. And thank you for the change in venue. I wanted to start by welcoming everyone for joining us today, including our representatives from Alzheimer's LA. I also wanted to thank the Department of Neurology for allowing us to utilize one of the Wednesday morning slots that's usually allocated for grand rounds. So thank you to that. I want to also acknowledge those who helped to organize this event. So before we get started with the good science and the exciting innovations we're going to hear about, I want to make sure to thank the staff. So Nancy Osich has been organizing this for many years and critical in putting this together. Dorothy's not in the room, but I want to thank Dorothy Lee for helping to organize as well. And our UCLA development colleagues are not here as well, but they're critical in putting together events like this and interacting with our generous donors. So thank you. This is now the 35th year of the Turken program, as Dr. Carmichael alluded to, and we're about to celebrate, for reasons that you can ask me about later, our 37th Turken awardee. And it's a unique opportunity for our community to come together and center around the idea of neurodegeneration in all its forms, exchange ideas, and advance new thinking in Alzheimer's disease and related dementias. So I won't echo the comments that Tom said about what an exciting year it's been in the last couple of years in the field with new biomarkers and new treatments, but simply to say that these treatments we recognize are not the end solution to the variety of pathologies that result in dementia. This progress will require us to explore and develop new pathways, really question what the core assumptions are in the field, and then pursue those towards defining mechanistic insights. And so that's the spirit of what the Turken Foundation wants to support. And it's typically manifest in young individuals, bright scientists who bring those new ideas to the forefront. It's also this unique opportunity here at UCLA to bring the neurodegenerative community together. And right now, that's been a very exciting time in the last couple of years. We're working to drive advances in incidence and risk detection, biomarker development, diagnostic precision through understanding genetic risk, and importantly, therapeutic discovery. And while we do that, we're trying to do it in the pursuit of delivering equitable care across the spectrum. So this is an opportunity both in the lectures today and then in the abstract presentations later and the poster session that will follow, an opportunity to recognize all of those contributions and the collective effort that we're all putting in. I also want to take a moment just to thank our partnership with Alzheimer's LA. This is now a long-standing partnership that is catalyzed by the Turken Foundation and through our donor, Beth Devermont. She's a longtime benefactor to UCLA and the Easton Center and sponsor of this award. And she's unable to join us today due to illness. I'll read some remarks that she provided a little bit later. And finally, today's agenda highlights the important progress that we are making towards a mechanistic understanding of the tau protein and its role in Alzheimer's disease and related dementias. And this, I think, will be the defining characteristic of the next generation of therapeutic strategies that target this problematic protein. So with that, I'm going to introduce last year's awardee so we can hear an update from Dr. Chao Peng. Chao is an assistant professor of neurology, and he's going to present the progress from last year's project, Transmission and Conformational Diversity of Pathologic Proteins in Neurodegenerative Diseases. Thank you, Chao.
Yeah, thank you, Jason. And it's kind of really exciting to come back this year and show the progress in our lab. So my lab really focused on the transmission and conformational diversity of pathological proteins. And as we all know that most of the neurodegenerative diseases are characterized by the accumulation of pathological proteins in the brain. For example, Alzheimer's disease is, For example, Alzheimer's disease is characterized by the accumulation of A beta and tau, and the Parkinson's disease is characterized by the accumulation of pathological synuclein in the neuron. So my lab really focused on tau and synuclein, these two pathological proteins. During the past 2 decades, there is more and more evidence to support that many of these pathological proteins have some prionic features. For example, pathological synuclein could use itself as a template to recruit the soluble synuclein in the brain and change its structure from this normal structure to a pathological structure. And by repeating this process, pathological synuclein is able to amplify itself and eventually form this huge protein complex as we regularize the Lewy bodies in the cell. And what is even more interesting is that this pathological protein, they could transmit from one cell to the next cell and repeat this amplification process in the recipient cell. So which is believed to be highly correlated with disease progression. And the mechanisms that regulate this transmission process is a hot topic in research. The reason is that if we can find a way to reduce or block this transmission, we basically can potentially reduce disease progression, which could be a therapy for these diseases. So my lab will also focus on these like the molecular mechanisms that regulate the transmission. This is a review I read for the field about several years ago. And when writing this review, what I noticed is that when people start this transmission, they kind of focus on the pathological seeds by themselves, right? How the seeds are released, how they are taken up, how they are degraded. However, if you look closely for the pathological seeds to amplify itself, you also need the involvement of the monomer, the soluble protein in the cell. For example, for pathological synuclein to amplify, you need the soluble synuclein as a substrate for amplification. So the question that we try to ask is that whether this kind of regular soluble synuclein, could they play a regulatory role in the amplification of the pathological synuclein? So there are many PTMs, like post-translational modifications on soluble synuclein that has been reported. For example, phosphorylation site, cetynation site, ubiquitination site. And we kind of ask whether when soluble synuclein carry these PTMs, will that change their interaction with pathological synuclein and eventually change the amplification of pathological synuclein. So we built a cell model to test this. This is 293 cell. We express like mutant synuclein carry like different mutations to mimic phosphorylation site on synuclein as a platform to test the seeding. So you can see we use tyrosine Y2E or S2E mutation to mimic either tyrosine or serine phosphorylation. And then we train these cells that express this mutant synuclein with pathological seeds. In this particular case, we use PFF, which is the artificial generated pathological synuclein. And you can see that we really don't see much effect actually for this different mutation on the amplification of PFF in the cell. However, we all know that this artificial generated pathological synuclein has a different structure compared with the real misfolded synuclein diseased brains. So we repeat the same experiment, but using soluble pathological synuclein we purified from PD brains. So this is what we call Lewy body synuclein. And you can see now we see a dramatic effect. For example, tyrosine 39 phosphorylation reduced the Lewy body synuclein amplification by about 50%, and S87 phosphorylation really increased the Lewy body synuclein amplification. And this kind of demonstrates two things. Number one, soluble synuclein phosphorylation indeed modulate pathological synuclein amplification. Number two, the effect seems to be pathological synuclein conformation dependent. So previous study has shown that, like including ours, showing that pathological synuclein could form different structures in the diseased brain. I want to draw your attention to this disease called multiple system atrophy, because in this patient, pathological synuclein accumulated in glial cells instead of neurons, and we call it glial cytoplasmic inclusions. And we know that this GCI synuclein have a different structure compared with Lewy body synuclein. So then we performed the same experiment, but using this GCI synuclein, which is the pathological synuclein from MSA patients, and to see whether soluble synuclein PTM could also modulate GCI synuclein amplification. And you can see that very clearly, this phosphorylation site could change the amplification of GCI. But most interestingly, if you compare the data from PFF, Lewy body, and GCI together, you see a very different pattern. For example, S87 phosphorylation increased the Lewy body synuclein amplification, but very dramatically reduced the GCI synuclein amplification. That really means that the effect of the soluble synuclein PTMs is highly conformation dependent. So because previous study of synuclein PTMs has been really focused on the PTMs on pathological synuclein, there's no kind of systematic identification of PTMs on soluble synuclein. We basically build a protocol to generate highly purified soluble synuclein from diseased brains, and we performed a very deep mass spectral analysis to identify all the PTMs on soluble synuclein in diseased brains. And you can see here's a summary of the result, that we identified a lot of lower PTMs, and this is a breakup view of the data that to show which PTM is identified in what type of disease. I want to show you that we actually identified a lot of interesting lower acetylation sites on soluble synuclein. And then we ask, okay, is the effect of synuclein PTM restricted to phosphorylation, or does acetylation on soluble synuclein could also change the amplification of pathological synuclein in diseased brains? So we did essentially the same experiment, but we use K2Q mutation to mimic the different acetylation on soluble synuclein. And you can see that we get a very similar conclusion that this acetylation site could change the amplification of pathological synuclein. And if you put the data together, you could again see that the different synuclein conformation has totally different responding pattern to soluble synuclein PTMs. So all this data is done in 293 cells. Finally, we try to kind of replicate the experiment to verify whether we see a similar thing in primary neurons. So we culture primary neuron, infect them with either wild-type synuclein or S87E mutant synuclein, and then we induce pathology in these neurons using GCI, Lewy body, or PFF. And you can see we get a very similar conclusion that S87E phosphorylation really blocks GCI amplification, but increased the Lewy body synuclein amplification. So hopefully, so far we have convincing you that soluble synuclein PTM is indeed a very critical regulation mechanism to regulate pathological synuclein amplification in diseased brains. So during the past year, with the support with the Turken Award, we actually expanded this study to the tau field. So the question is, that soluble tau PTMs also regulate pathological tau amplification, particularly in AD brains. So to do that, we built a platform to purify soluble tau from diseased brains. And with this highly purified tau, we could perform mass spectral analysis on PTMs on soluble tau. And you can see that, I want to say that all of these are unpublished data, but you can see that we identified tons of novel PTMs on soluble tau. We counted over 100 new PTM sites that we identified. This is a breakup view, and you can see we did AD, CBD, PSP, Pick's disease, and normal controls. These are all different tauopathies, and it shows different like PTM pattern. And then we want to ask, does these PTMs on soluble tau also change the application of pathological tau in these brains? So build a cell model that you can express mutant tau in 293 cells, and then for this particular experiment, we focus on acetylation. So we use K2Q mutation to mimic the different acetylation sites on soluble tau that we identified. And then we purified pathological tau seeds from AD brains and use those seeds to induce the tau aggregation in the cells. And then we quantify the amount of pathology induced to evaluate how this acetylation site will change the amplification of pathological tau in AD. And you can see the data is summarized here, that indeed we find some of the acetylation site, like K280 acetylation, which is in the microtube binding domain, increase AD tau amplification, but K370 acetylation, which is on the C terminal, reduce AD tau amplification. Very interestingly, if we do the same experiment, but using pathological tau purified from CBD brains, as you can see from here, we don't really see much effect at all. And if you combine the two data together here, you can see that clearly AD tau and CBD tau shows a very distinct pattern in response to different acetylation site on soluble tau. And then we did the same experiment for phosphorylation. So for phosphorylation, we kind of separate the three groups. It's either serine phosphorylation, serine phosphorylation, or tyrosine phosphorylation. So for serine phosphorylation, we test 4 of them, and you can see that the two serine 212 and 217 phosphorylation, which is in the proline-rich domain, interesting, increase AD tau amplification, but the C-terminal phosphorylation, the negative binding domain phosphorylation, the surrounding 361 phosphorylation, very dramatically block AD tau amplification. Which also demonstrated that phosphorylation can modulate AD tau amplification. And then we did the serine phosphorylation, and we also tested 4 different serine sites. And very interestingly, all these serine phosphorylation don't affect either AD tau or CBD tau amplification at all. And you can see they all don't show any effect. But very interestingly, I want to draw your attention to this site for the serine 214, because surrounding 212 and 217 phosphorylation both increase AD tau amplification. And the serine 214 is kind of right in the middle, and these sites don't have much effect. So that really suggests that the location is not that important, but the type of amino acid actually determine whether the phosphorylation will increase or decrease or have low effect on AD tau amplification. And we also did tyrosine 21 phosphorylation, and that increased AD tau amplification a little bit. So if we combine the data together, you can again see the totally different response for AD tau and CBD tau to soluble tau phosphorylation. And then we ask, because for acetylation, we know that some of the acetylation sites, like 280 acetylation, increase AD tau amplification. Some of them, like 370 acetylation, decrease AD tau amplification. Then the question is, if we have multiple acetylation on the same molecule, which one will dominate it? So we first test whether the acetylation sites that are next to each other, will that increase the effect? So you can see 280 acetylation increase AD tau amplification, as we know. But if you add on top of that 281 acetylation, that actually further increase the effect of 280, even though 281 acetylation by itself don't change AD tau amplification at all. So that means there's a synergistic effect. And we do the same thing for K370 acetylation, which shows to reduce AD tau amplification. And you put on top of that 369 by itself don't reduce AD tau amplification, But you put them together, the effect is kind of similar to 370. That means these two sides, 369, don't really contribute much to the effect on this region. So then the most exciting part is to now, if we combine the 280, 281, which you can imagine as the bad acetylation, with the 369 and 370, which you can imagine as good acetylation, which one will dominate the effect? So you can see 280, 281 increase A-tau amplification, but if you just put 369 a further acetylation on this molecule, that completely abolish the effect of 280 and 281 acetylation and reduce the amplification even lower than the wild-type tau. And 370 could do the same thing, and you combine those two together, again, shows similar effect. That means if you have this bad acetylation in the molecule that increase A-tau amplification, even if you can put one protective acetylation site, you kind of could abolish this toxic effect and have a potential preventive effect on A-tau amplification. Again, we don't see much effect for all these combinations for CB tau again. So next, we want to ask, what is this done in cell? Do we see similar effect in mouse? So we basically overexpress the mutant tau or wild-type tau in the hippocampus region in the tau local mice. And then we inject the mouse with AD tau that we purified from AD brain to induce pathology. As you can see, these different mutations or the wild-type tau have similar expression level. But when we use AD tau to induce pathology, you can see it induces much more pathology in the K280, 281 mutant tau, and much less pathology in K369 and 370 mutant tau, which again demonstrate our effect in vivo. Finally, we use like modeling to see whether we can predict this effect. So the cryo-EM structure of AD, CBD, and PSP are all known. So we just evaluate whether this mutation will change the energy of this fiber structure. And very interesting, you can see for most of them, the energy change is either negative or close to zero. That means they kind of stabilize the structure. But just for K370, that really increased the energy of AD tau fiber, which is what we see, why we see that is prevented. We did the same thing for PSP tau, and it seems that there should be no effect of these mutants on PSP tau amplification. And finally, we use the experiment to verify that indeed this phosphorylation site don't affect PSP tau amplification. So with that, I would like to summary that we basically find a lower mechanism that regulates both pathological synuclein tau amplification, which is the soluble synuclein tau PTNs. And I would like to thank my lab members who are directly involved in this work, and also my collaborators and funding resource. Thank you so much.
We have time for just one question for Dr. Peng. We have a microphone, 1 second for our Zoom colleagues. Dr. Cole.
So just to relate this to therapeutics, people have tried to use, like Ligan has tried to target acetylation at UCSF for many years and had trials with salicylate. So you're trying to control the acetylation, deacetylation. Most of those things seem to be less specific than what you're talking about. And then what you're also saying is that depends on which tauopathy. So most people go and say, I'm going to just test PSP because it's an orphan disease and it's pure tauopathy. So can you comment on the clinical relevance of your findings? I think they're obviously important, but for the broader audience.
Yeah, that's a very perfect question. I think right now one of the challenges that the knowledge we get for tau acetylation on its amplification also is mainly used like recombinant proteins or artificial systems. So what we find is that like to really use the human disease-related conformation is very critical to illustrate the effect of soluble tau acetylation. So I think there's still potential. Obviously, these things will be, which I don't think we are able to find a reagent that will specifically target tau. But I think all the effect could be balanced by the concentration or how you deliver the drug. So we still think that it's possible to identify compounds that modulate the soluble tau acetylation that could reduce tau amplification, which could be a good start point for clinical trial. We actually already have some of those data that I don't get a chance to try, but yeah, we do see a very strong effect with some of the compounds we tested that could block A-tau application.
Great. We have to stay on schedule so we can get to all the talks. Dr. Peng will stick around and be available, I think. Thank you, Chao. And that was really an excellent presentation and clearly indicates why you were last year's award winner. So thank you very much. At this moment, I think I'd like to introduce Jennifer Schlesinger, who is the Vice President of Healthcare Services and Professional Training from Alzheimer's LA, who's going to make a few remarks. Thank you very much, Jennifer.
Good morning. Thank you, Dr. Hinman, and thank you for that wonderful presentation, Dr. Peng. Alzheimer's Los Angeles's mission is to provide care and support to families who are affected by Alzheimer's disease and related dementias. And this mission goes hand in hand with the longstanding commitment of Beth Devermont, the Sam Ida Turken Foundation, and the Mary S. Easton Center at UCLA to support the careers of early researchers and hopefully through you, to find a cure and prevention to this devastating disease. When we work with families, we talk to them about we can't modify the disease trajectory for them at Alzheimer's LA, but what we can do is change their experience with the disease by providing them with support, by providing them with tools and strategies to navigate the disease, by providing them with compassion and helping them understand that they're not alone. But what's so exciting to us at Alzheimer's LA and to our community at large is you are changing the trajectory of the disease through your work. And that's what provides our families and that's what provides our communities with optimism and hope for what's ahead. We deeply admire all of you, the researchers who are advancing the science and are finding these reasons for optimism. I want to congratulate Dr. Avi Samelson and all of the researchers here today. We look forward to learning more about your work. It's because of your tireless devotion that we give our families hope. And it's through this long-standing relationship that we're able to hold this important space together and to elevate what I think is just an amazing brain trust that we have here. So thank you to my colleagues at UCLA. Thank you to Beth. I'm so sorry that she's not here today. I know she loves coming and learning and seeing everyone, but we're so grateful to the Turken Foundation. And of course, again, congratulations to Dr. Samelson.
Thank you, Jennifer, and we're delighted and honored to have you as a co-sponsor for this event. As Jennifer mentioned, and as I alluded to earlier, Beth Devermont couldn't be with us today due to illness. She did send me some comments, and those comments relate to today's event, but also to her leadership of the Sam and Ida Turken Charitable Foundation, which is supported in memory of Beth's mother, shown here on the slide. I'll just read her remarks. I know it's unfortunate she wasn't able to be with us because she brings an energy and passion for meeting the awardees and engaging, hearing about the progress. And so it's very unfortunate she wasn't able to join us. Here's what she asked me to read. Dr. Samelson, 2025 marks the 35th year the Turken Award is being presented. While I'm sorry I'm unable to be with all of you this year, our mission remains the same, and that is to give a tool in the form of a monetary gift to an early career researcher in Alzheimer's disease or related fields to continue and deepen their work in the field. We believe our awardees will contribute something significant in our quest to prevent or cure this disease and hope this will remain their career path for all of their working years. Dr. Samelson, for this reason, we are very honored that you have been chosen as this year's awardee. As I will be getting a recording of today's program, I will be able to hear your presentation of your work, as well as that of Dr. Peng and Drs. Hu and Han. I look forward to having an opportunity to discuss your work with you and plan to reschedule our regular post-event lunch later this month. My best to you, Beth Devermont. So that's very exciting. Let me go forward. I'll take this opportunity to highlight the lengthy list of awardees who have received the Turken Award in past years. Many will note colleagues amongst the audience, which is an important recognition of the continual support of this activity here at UCLA. Many will also note that there are significant names in the field who have made meaningful contributions to date. So the awardees who are more recent will note that they have large shoes to fill as they join this somewhat distinguished list. The committee had a very difficult time with quite a few important and impactful nominations from junior scientists and physician scientists here at UCLA. But the nomination of Dr. Samelson won out. So with that, I'd like to introduce Dr. Samelson's nominator, Dr. Lawrence Sapersky, Distinguished Professor in the Department of Biological Chemistry at UCLA, who nominated Avi. Thank you, Larry.
Good morning. So it's a pleasure to introduce Avi. Avi's work focuses on understanding protein folding in both normal and pathological conditions. As a graduate student at UC Berkeley, Avi developed ways of studying protein folding during translation, which was an important departure from traditional ways of studying protein folding, which was typically in dilute solutions, and arguably this was a more closely related way to study how proteins fold in vivo. These methods allowed him to establish principles of co-translational folding, ensuring both normal protein function on one hand and preventing misfolding on the other. Misfolding, of course, is a central feature of many neurodegenerative diseases. As a postdoctoral fellow, Avi joined Martin Kampman's lab at UCSF to use a genetic approach to understand mechanisms of tauopathies, a major class of neurodegenerative diseases. Avi's approach is compelling. It is remarkable, and I would say totally unexpected extension of molecular genetic approaches that attracted me to molecular biology many, many decades ago, using approaches that were pioneered by Sidney Brenner and Francois Jacob in their efforts to genetically dissect complex cell biological pathways. And they used as their proof of principle DNA replication, and they carried out genetic screens looking for mutations, conditional mutations that disrupted DNA replication, and used this really as a framework and starting point for understanding the biochemical pathways that regulate this process. This approach was powerful and was adapted to study cell cycle regulation in yeast, to study early embryogenesis in Drosophila, and to dissect cell death pathways in C. elegans, and to take on and dissect many other fundamental processes in biology. With the discovery of single gene mutations leading to neurodegenerative diseases, geneticists studying model organisms realized that they could introduce these mutations into, for instance, flies and worms, that these cause neurodegenerative phenotypes, and that they could then be used to model, as models to screen for mutations that modified the phenotypes, thereby identifying candidate genes and processes regulating neurodegeneration. Ideally, of course, one would like to have done these studies in human neurons. And with Yamanaka's discovery of human-induced pluripotent stem cells, or iPS, cells as an experimental system, and the advent of CRISPR technology, it became possible to modify iPS cells to express disease alleles, to phenocopy neurodegenerative diseases, and then to carry out high-throughput genetic screens to identify modifiers of this pathway. Avi has used this approach in a spectacular way. He's identified expected and unexpected modifiers and pathways, providing genetic and biochemical entry points to better understand tau proteostasis and to provide new therapeutic approaches for tauopathies. And it's this exceptional work, a particularly creative and elegant adaptation of classical genetic modifier screens, that Avi will tell us about today. The title of his talk is New Regulators of Tau Proteostasis, revealed by CRISPR-screening of human neurons.
Great. Thank you, Larry, for that very, very, very kind introduction. I'm really excited to be here and to have been nominated for this award. And I'm excited to tell you about my work trying to understand protein aggregation in Alzheimer's disease. Okay, but before I start, oh, that didn't work. Okay, are we ready? Okay, so before I start, I just want to thank the Sam and Ida Turken Charitable Foundation and Beth Nevermont, first and foremost, for this award. Second, I want to thank Tom, Larry, and Jason for both recruiting me here, but also for continuing to make the neuroscience and the neurodegeneration communities here as strong as possible. And then finally, I just want to thank the Department of Neurology admin suite, who has been just incredibly helpful for helping me get my lab going from when I arrived here in January. Okay, so let's now talk about science. Okay, so as I'm sure all of you know, Alzheimer's disease is a progressive neurodegenerative disease that affects about 7 million Americans, and by 2050, that number is expected to double. And a defining pathological hallmark of Alzheimer's disease is the aggregation of the protein tau in neurons in the brain. And tau aggregation is correlated with neuronal death as well as cognitive decline. So what I'm showing you here is the amount of aggregated tau as measured by PET imaging in human patients. And what you can see is that aggregated tau is basically negatively correlated with neuronal metabolism, which is a proxy for just how many neurons are there. And in addition, it's negatively correlated with basically cognitive tests. And this is kind of in contrast to A beta, which shows much weaker correlations in disease. Okay. So in the past 10 or 15 years, we've come to understand that tau aggregates are not just amorphous blobs of proteins sitting in neurons in the brain. They're these really incredible and highly ordered structures. So this is a cryo-EM structure of a Alzheimer's disease tau aggregate. And what I'm showing you here in this cartoon is just the view down the axis of this fibril. So this is the ordered part of, and cryo-EM also has revealed that not only are these aggregates ordered, but they're also their structures are actually disease specific. And this kind of poses a paradox or a contra-dogmatic observation for the way we normally think about how protein acquisition occurs. And generally, the way we think about how protein acquisition is organized is that the amino acid sequence of the protein gives us one three-dimensional ordered structure. But that's clearly not what's going on in neurodegeneration. And I think it's just kind of a central mystery for the next 10 years of AD and related dementia research. Okay, so what is controlling this kind of incredibly fascinating property of this protein? So one clue is just from the pathology of tauopathies themselves. So just as an example, if you look at Alzheimer's disease and another tauopathy, Pick's disease, they have two different three-dimensional structures, but they arise in different regions of the brain. So what these cartoons show is basically the initiation and spread of disease. So darker color means this is where aggregates first appear, and then they spread to the lighter colors. And I hope what you can see is that these colors are totally different. even though they involve aggregation of the exact same protein. And what this tells us, I think, is something very fundamental, which is that the cellular environment is controlling this organization process of tau into aggregates. And understanding how the cellular environment controls this process is really the goal of my research. So as Larry said, I come from a protein folding lab. And when we think about protein folding, we think about trajectories, or what are all the conformations that a protein must populate to reach their final structure. And I think of aggregation in the same way. And what my lab is really interested in is how cells control that trajectory from a non-pathological monomer through prefibrillar oligomeric states, and then to the final fibrils that we see in human patients. And obviously, the idea for understanding this process is maybe we can intervene at different control points during this process. Okay, so my lab uses iPSC-derived neurons, where we can take fibroblasts from patients, turn them into induced pluripotent stem cells, and then differentiate them into neurons. And the reason why we like to use this is because these are human cells. They're neurons. They, for instance, endogenously express tau, which is not true of many other cellular models. And importantly for me, as kind of coming from a biophysics world, is we're retaining these proteins in their native environment of a human neuron. And we arm these iPSCs and iPSC-derived neurons with machinery to do to manipulate gene expression using CRISPR. And we have a number of different CRISPR systems, but today I'll just introduce two. So how this one called CRISPR-I works, so that stands for CRISPR interference, is we take a dead Cas9 molecule, so it cannot cut DNA. We fuse it to a transcriptional repressor, and then when we load it with the guide RNA, it goes to a region in the genome, and it directs this repressor to then turn off gene expression. We can do the same thing by fusing, instead of a repressor, an activator in CRISPR-A, or activation mode, to induce gene expression. And what this enables us to do then is take many, many guide RNAs targeting many, many genes and use them all at the same time, so that we can, for instance, assay the entire genome in one very large scale experiment. And as a postdoc, I was really interested in understanding the first stages of tau aggregation. It's transitioned from a monomer into these prefibrillar oligomers. And oligomers, in addition to being the very earliest stage of aggregation, are thought to be more toxic than these end-state fibrils. And so the question that I asked was, can we systematically determine how human neurons control this conformational change? And to do this, I conducted the conversion PowerPoint. And to do this, what we did was do a genome-wide CRISPR-I screen for tau oligomers. And the way we do the screens, we take our CRISPR-I iPSCs, we infect them with guide RNAs against every gene or protein coding gene in the genome, differentiate them into neurons, and then we let the neurons mature, fix and stain the neurons with an antibody against tau oligomers, and then we just use flow sorting to sort our neurons into those with high and low levels of oligomers. And then we use next generation sequencing to sequence those two bins of cells in order to call which genes control tau oligomerization. And when we completed this screen, we got over 1,000 hits. And just to orient you on this plot, genes in pink, knockdown of those genes increase oligomer levels. Genes in light blue, knockdown of those genes decreases oligomer levels. And we assign a p-value to each gene, which is our y-axis here, by comparing each gene to thousands of non-targeting control guide RNAs that are in the middle in black. And This was really exciting when we finally got all of the data back. We used gene set enrichment to classify genes based on type, which is what these kind of bolder colors are. And we found many new pathways, as well as a lot of expected pathways, which made us confident that this assay actually worked. But today, I'm just going to talk to you, tell you about one gene, which is this gene Col5. Okay, so what is Col5? So Col5 is a E3 ubiquitin ligase, and the role of ubiquitin ligases in cells is to tag substrates, which I'm showing you here in blue, with a molecule called ubiquitin. And when substrates get polyubiquitinated, they generally get sent to the proteasome for degradation and recycling of their constituent amino acids. So Col5 is a special type of E3. It forms colon ring ligase complexes, which is the CRL. And generally, the way these complexes work is the colon itself, which is in purple, Col5 here, serves as a scaffold for an E2, which you can see on the right, and the substrate. And substrates are bound using modular substrate-specific adapter sequences that recognize degrons, which are short amino acid segments that the protein recognizes for ubiquitination. Okay, so the first question we wanted to ask was just, how does Col5 control tau levels? So we built a dual fluorescence reporter, which I'm showing you here on the left, where we fuse tau to GFP, and from that same transcript, we then express a red fluorescent protein. So by measuring the GFP to MAPL ratio in our iPSC-derived neurons, we can ask how do changes, for instance, Col5 knockdown, in the cell lead to changes in tau levels. And when we do our Col5 knockdown, which you can see on the right here in blue, we saw an increase in tau levels, which recapitulates our screen phenotype, and this doesn't occur with GFP, which is our control. And when we saw these data, there are basically two hypotheses that we considered. One is tau is being directly ubiquitinated by Col5, or there's some other indirect effect. And to test which one of these might be the case, we thought that if we inhibit the proteasome, if there's indirect effects going on, we probably won't have a strong effect on Col5-mediated increases in tau levels. But if it is being directly tagged with ubiquitin, we should see a pretty drastic change. So when we inhibit the proteasome with this drug called MG132, you can see that this increase in tau levels due to Col5 knockdown is totally reversed. And this was the first suggestion that, okay, we might have found actually a new tau E3 ubiquitin ligase. So if this is a true E3 for tau, there should be a degron. And so we cloned 50 amino acid fragments of tau into this same reporter and reassayed the entire protein to try and narrow down where the degron is. And when we did this experiment, we really only found one region of the protein that was responsive to Col5 knockdown, just like full-length tau is in blue, and that's this region 80 to 130 in the tau sequence. Okay, so we're starting to kind of build up a model where now we think tau is the substrate for this complex, and the complex recognizes residues 80 through 130, but we're missing a big piece, which is this substrate adapter. And this is kind of the beauty of being able to do genome-wide genetic modifier screens, is you just go back and look and ask, of all of the known Col5 adapters, which ones recapitulate our Col5 screen phenotype? And really the only one that did is this gene called SOX4. And when we overexpress SOX4 using CRISPR-A now, we see a decrease in tau levels. So this rationally makes sense. If you have higher levels of the adapter that brings tau to the complex, you should increase ubiquitination and decrease tau levels. And when we reconstitute this system in HEK cells, we see the exact same thing. Additionally, we were able to test for if expression of SOX4 increases the amount of ubiquitinated tau. And I'm sure what I'm showing you here looks kind of like a mess, but just take a look at these lanes in blue. So if we overexpress SOX4, or in this lane, empty vector, and look for ubiquitinated tau, which is shown here, you can see there's a pretty big increase in the amount of ubiquitinated tau here, and not when we do this with GFP. Furthermore, if we inhibit activity of colons with a pan-colon inhibitor called MLN4924, you can see that the amount of ubiquitinated tau decreases. Finally, we reconstituted the entire ubiquitin cascade in a test tube with all purified components. And I'm just going to draw your attention to these two lanes. So if you reconstitute the entire cascade, but you don't have SOX4, you can see there's no polyubiquitinated tau in this lane. But when you add SOX4, you get this nice streak, which represents tau being polyubiquitinated by the colon complex. And I think the combination of these experiments showed that SOX4 is both necessary and sufficient for ubiquitination of tau. Okay, so here again, we've built our model. We have a CRL5 SOX4 complex. It ubiquitinates tau and recognizes tau with a degron that is here. And one implication of our identification of this complex is that perhaps enhancing this interaction or the activity of this complex could be used just to lower tau levels as an AD therapeutic. But all the experiments that I've shown you so far have just been in a dish, although they are in iPSC-derived neurons. So we decided to collaborate with the Allen Institute to take a look and ask a few kind of just basic questions. So what does COL5 expression look like in Alzheimer's disease? And how does it, and is it related at all to vulnerability or resilience of cell types in the brain? So the plot I'm showing you here is a graph of basically resilience in AD. So dots on the left mean that that cell type is more vulnerable in AD. There's more death. And on the y-axis is just COL5 expression. So each dot represents a cell type in a human brain. And what you can see is that we have a moderate but statistically significant correlation of COL5 expression with AD resilience. So the more COL5 you have, the less death there is in AD. And this is also true for this accessory protein called Ariadne, which I didn't really talk about today, but is essential for COL5 function, as well as our adapter SOX4. And we also wanted to look if this was true across some other tauopathies. So in collaboration with Jessica Rexak here at UCLA, we looked at the correlation of COL5 expression, as well as every single COL5 adapter, which I'm showing you here in blue, with a single cell trajectory of vulnerability to resilience across three tauopathies, FTD, PSP, and AD. And what we found was not only was COL5 highly correlated with that trajectory, as were a number of different COL5 substrate adapters, but they were more highly correlated than some of the traditional factors that we think of as protective in Alzheimer's disease, like the protein VCP here in black, or CHIP also in black. Okay. So just to summarize, we've found one protein on this pathway that I talked about today that we think stops tau oligomerization via basically a housekeeping function by degrading tau. And we also found that COL5 drives resilience, or is correlated with resilience in Alzheimer's. And we're really excited to follow up kind of three hypotheses to further understand how COL5 is related to cell type specific resilience in AD, including maybe it's just about tau ubiquitination. Perhaps it's also perhaps Col5 also is degrading some vulnerability factors. And then our third hypothesis to try to answer this question is that maybe there's some downstream indirect effects of Col5 activity. And with my last 30 seconds, I just want to mention our ongoing projects in the lab. So one of our major goals is to characterize the function, the structure and function of this complex with the goal of enhancing the interaction for a potential therapeutic. And Manasi Mova, a great second-year graduate student, already has some pretty exciting preliminary data showing tau and SOX4 binding using NMR. Additionally, we're more generally interested in how sequence controls aggregation, and the postdoc in the lab, Shraddha KC, is adapting some of the high throughput technologies that I created as a postdoc to understand how sequence affects aggregation. And then one of the third goal of my lab is to create better models of neurodegeneration in a dish. Sandy Rashed has made some really awesome progress making inducible systems so we can do gene knockdown that doesn't affect differentiation of neurons and things like that. So we can deconvolute effects with our aggregation phenotypes from kind of neurodevelopmental phenotypes. So with that, I'd just like to thank everybody who's involved in this work. So these are all of the people in my old lab and our collaborators. And then I'd like to thank my lab, our current collaborators, the 5th floor of MRL, and funders. And again, I'd really like to thank the Sim and Ida Turken Foundation for this award. Thank you so much, and I'll take any questions.
Thank you, Dr. Samelson. I think we can see why you're deserving of this year's award. Any questions for Dr. Samelson? We have a couple minutes in the back.
Hey, Avi, this is a great talk. I have a slightly more upstream question. So let's say tau gets into the neurons either by HSPGs or in LRP1 or whatever. Presumably, it's still in these vesicles or some sort of endosomal body. Is all of this happening once it's released into the cytosol, or is it happening at like multiple places? How does it get shunted or shuttled to all these different bins?
Okay, that's a great question. So I think the question is, when tau gets uptaken by a neuron, what does your screen have to do with that? Basically, my screen has nothing to do with that. This is not a screen in a seeding paradigm. This is endogenous tau just at baseline. So these hits will not tell us anything about uptake. There's no seeds here. However, we did collaborate with Lee Gann's lab at Cornell to do a seeding based screen, also in iPSC-derived neurons. And there, all of the factors that you would expect come out, like VPS13, a bunch of endolysosomal system things, and are like super strong modifiers that take that increase aggregation. So yeah, and also obviously it depends like on the mechanism that you do this experiment with, right? Different types of tau seeds get into the cell in different ways.
Yeah.
So that'll bias you to how to what genes you find. So yeah, we're excited to do some of that. But if I go back to me, it's if you go back here, the seeding, right, is like way down the line here, and kind of what my lab is interested in is this as like a protein folding problem. So like, how does this arise without a seed in some ways, which I know is kind of less exciting for some people. But as a protein folder, this is kind of what we're interested in. Okay. Jess.
SOX pathway. Oh, hello. What do you think about the pathway for druggability? And then why do you think that there's this heterogeneity and basically like an absence of it where it needs to be in disease? What are maybe the mechanisms or upstream aspects of that?
Okay. So I think the question is, first, what's the promise for druggability of these complexes? And then second is, I guess I didn't quite understand.
If the resilient cases have it, why is it there in the vulnerable cells? What happens to?
Oh, okay, yeah. Okay, okay. So to answer the first question, I think as a target, this is really exciting, and I'd say that there's like a lot of work to do to actually show that it is a real target. But it's exciting because these types of complexes generally have very weak binding to their substrates. So I think that there's a ton of room to improve binding. Now, you want to improve binding, but you want to do it in a specific way, right? Because if you are binding tau to an E3 ligase and it can never come off, then what you've done essentially is kill the complex, right? So I think that there's some really interesting like binding kinetic modulation that you would have to do, right? So you want to have like a really strong on rate, but then you want it to come off really fast. But I think that because these are usually weak, there's a lot of room to improve, which makes me excited. Okay, then the second question is, why does Col5 go away? So I think there's two things. One is if you stratify by cell type, at least in CAD, many of the vulnerable cell types they have low Col5 expression at BRAC 0. So they start out with low expression. But in some of the other data that we've looked at together, you get downregulation in Col5, and downregulation of E3 ligases is a signature in SST neurons too in early AD. Okay, so what could be causing that? Just to kind of BS, it's possible that there is just like a general, maybe it's a signature of the cell's like death pathway. They're just like, I give up, kind of. I know very little about the regulation of these proteins at the transcriptional level. And this is something I'm proposing to do in a grant because the CRISPR systems are like perfect for doing these types of readout of experiments to find what is actually controlling Col5 expression and adapter expression in different stress contexts. I'm sorry, I don't have a good answer. Yeah, definitely. Thank you. Larry. Wait for the mic. Sorry.
Thank you. So in the assay that you've developed, what is the level of knockdown you typically get from genes with RNA with CRISPR-I?
Yeah, it is very dependent. So the question is, what level of knockdown do you get with CRISPR-I? I'd say it's generally like 99.9%.
I'm sorry, you mean basically it's a null?
Yeah.
Okay.
Yeah. But the nice thing about the way we do these screens is there's 5 guides per gene. And so you actually get you get a titration of knockdown because some guides are not as good as others. So for essential genes, these screens can be really nice because you might have guides that knock down like 20%, or you have 20% expression levels of wild type. So you can
you get a series.
So you can reveal phenotypes without killing the cells, basically. Yeah, it's titratable.
And how many different E3 ligases are there in the iPS-induced neurons that you?
Hundreds.
Hundreds?
There's hundreds of E3 ligases, yeah.
And what sort of effort have you made to look at combinations?
To do combinations of ligases?
Of knocking out combinations?
Zero effort.
So could you speculate on the issue of redundancy and whether you're really just hitting the tip of the iceberg here?
Yeah, so Yeah, thanks for that question. Uh, yeah, so yes, there's tons of redundancy in these systems. For tau, we know that there's 3 or 4 E3 ligases. Tau is also degraded via autophagy. So that's why we see, that's I think why we see these small effect sizes, is because there's so many different ways tau can be degraded. Tau also can be degraded just via the ubiquitin independent mechanisms. So yeah, doing something like that would be a great experiment. And also, I'm writing a grant to do kind of like a genetic interaction map with COL5, and that's one way you could, for instance, unmask those types of effects.
Thanks.
Great, last question.
So given that there are a number of ubiquitin ligases that would turn over tau normally due to potentially just misfolding with its aging as it comes off of the microtubules or some other normal process, is there selectivity for misfolding with these different ubiquitin ligases that would make COL5 be recognizing an aberrant species of tau, preferentially possibly due to the PTMs described by Chen?
Yeah, thank you for that question. The question is basically, is there specificity to tau conformational state, which can be driven by PTMs, things like that. So for this system, we don't know. We hypothesize, so SOX4 has a domain that binds to phosphotyrosine. So we think that may be a consideration here. For other tau E3 ligases, so CHIP recognizes neo C termini that are created by caspase cleavage of tau. So we know that it definitely happens for tau and across the proteome.
Great. Thank you, Dr. Samelson. Stay here for a second. I'm going to present you with the official Turken Award for this year. Well, the Turken Day is, the Turken Symposium is primarily focused on providing the distinguished award. The other component that I mentioned is the bringing everyone in the neurodegenerative community together to share data and updates, and for me personally, reviewing the abstracts and making choices about these oral presentations is one of the more challenging components of leading the symposium activities today. There are so many good scientific projects happening that it was difficult to make decisions. However, we did come to 2 conclusions. And so we're going to hear from 2 abstract awardees who will present their work orally as a presentation. And this is one of the exciting components that will then transition to a wider poster session in the auditorium in just a few minutes. So with that, I'm going to introduce our first abstract awardee, who is Dr. Xia Han, a postdoctoral scholar in Dr. Jessica Rexach's laboratory. And Dr. Han will present her work on single-cell insights into young-onset Alzheimer's disease vulnerability. Thank you, Dr. Han.
Thank you to the Turken Foundation and thank you to the committee for this award. It's my pleasure to be here. I am excited to share our recent work in young-onset Alzheimer's disease and what we have learned about sex-specific and region-specific neuronal vulnerability. Young-onset Alzheimer's disease, or YO, are a group of people who develop AD before age 65, although they represent only 5 to 10% of all AD cases. It affects people at their working years, so cause major consequences in their social life, family, and work. So it raises a major health concern for the society. And we know mutations in PSEN1, APP, PSEN2 can cause some of the early onset Alzheimer's, but they just account for a small fraction of it. for most patients, we do not have the genetic cause. And unlike late onset Alzheimer's, which typically present with MSD-AD and have hippocampal involvement, young onset Alzheimer's usually have a higher cortical tau burden relative to hippocampus. And patients would present with atypical syndromes such as cortical basal syndrome, logopenic variant, which affect the superior temporal gyrus. And we know this type of hippocampal sparing AD is usually seen in males, while limbic predominant AD is more common in females. So there is sex difference. So in young onset Alzheimer's, there are many open questions. Do tau modifying mechanisms differ across these Yo syndromes, and what determines cellular and regional vulnerabilities? Working with our collaborators at Mayo Clinic, we assemble a Yo cohort that allows us to quantify tau burden across different brain regions, profile single-cell gene expression to identify what cell types are vulnerable or resilient, and we can also assess microglia activation pattern in response to tau pathology. So this is our data set. We perform single nuclear RNA sequencing across healthy control, the typical MSD-AD and 3 atypical AD syndromes, the logopenic variant, PCA, and CBS. We sample from 3 brain regions, hippocampal CA1, occipital lobe, and superior temporal gyrus. So they have distinct tau burden. STG is in logopenic, occipital lobe in PCA, and hippocampal CA1 is more affected in MSD-AD. So we have 140 samples from 70 donors in total. That is a good coverage for across region and diagnose. So we have annotated those major brain cell types, subtypes. We identify DEG, differential gene expression, and the gene regulatory networks. So specifically, we use a hippocampal specific AD dataset as reference to refine our neuronal subtypes. So we are able to identify region-specific neurons. So here are 4 hippocampal specific neurons, including CA1 pyramidal cells and dentate gyrus cells. We next ask whether these neuronal subtypes express marker genes that enrich for AD genetic variation, and how do they differ compared with late onset Alzheimer's. So we use LDSC to partition AD heritability. In young onset Alzheimer's, here we see 4 neuronal subtypes got significant enrichment for AD risk. And the strongest association comes from an excitatory neuron expressing neurogranin, then followed by a layer 2 and 3 cerebellum 2 positive neurons, and then a layer 3 to 5 ROBI positive neurons. And this enrichment is different from what we see in late onset Alzheimer's. So in load, although we see the neurogranin is still the strongest enrichment in AD risk, late onset AD specifically point to hippocampal specific neurons. So dentate gyrus cells and CA1 pyramidal cells are also enriched in AD risk. So here we see that genetic risk manifests in different cellular contexts in young onset Alzheimer's versus late onset Alzheimer's. Because we know that MSD-AD is more prevalent in females, and female might have hippocampal specific pathology. And there must be something that we have been thinking that for a long while, that something related to hormone decrease during menopause. So probably that we are curious to see how sex modify neuronal vulnerabilities, and especially when estrogen loss. So we look at estrogen receptors. So here, ESR1, it encodes estrogen receptor alpha, which is broadly expressed in the human brain and maintain the normal memory and learning process. So this gene is enriched in 3 subtypes. 2 of them are dentate gyrus cells and 1 microglia. And we found these 2 subtypes both show depletion in female versus male in hippocampal CA1. So it suggests that those female-specific vulnerable cells might be relevant to estrogen signaling pathway. To explore it, we identify transcription factor regulons using CINIC in each subtype and each brain region. So here I show the top 10 regulons in dentate driver cells, and here are the regulons ranked by their specificity in each disease. And we see several regulons activate in NSDAD are relevant to stress. So FOXO1 is triggered when estrogen loss. STAT1 is relevant to inflammation, and CUS1 maintains the neuronal identity. And FIA, we know it triggers an injury-like response. So the target genes of these regulons have a general upregulation in female versus male, which means female has triggered a stronger stress response than males in these female vulnerable dentate driver cells. And it is consistent with the hippocampal pathologies more common in females. And then in superior temporal gyrus, we see these two neuronal subtypes. Like I previously showed that these two subtypes are enriched in AD risk. One is the neurogranin positive cells, and another is the layer 2 and 3 cerebellum 2 positive cells. And here they both decrease in their cell proportion in NSAD and logopenic compared with health control. And there is a pronounced decrease in males logopenic. So these two AD risk neuronal subtypes can be male-specific vulnerable in this hippocampal sparing pathology. We also look at transcription factor regulons that activate in these two subtypes. And I found those regulons enriched for logopenic, also relevant to interferon response and inflammation and stress. Although we didn't see many differential gene expression for their target genes, we find one. PTPRN is the target of JUM regulon, and this gene supports neuronal resilience and is only downregulated in males in logopenic, which means that males might experience a weaker stress buffering. So these two neuronal subtypes in males might be less able to cope with AD stress that triggered by those AD genetic variations. So, which might explain why males have more hippocampal sparing pathology in this disease, in this region. So in short, we have demonstrated sex-specific vulnerabilities in young-onset Alzheimer's, that we have identified the one female-specific vulnerable dentate driver cells, which can be relevant to estrogen signaling dysfunction. In superior temporal gyrus, we find two neuronal subtypes that are male-specific vulnerable, and they are relevant to the activation of stress-related regulons. So this sex-specific and region-specific vulnerable neurons might partially explain, the hippocampal sparing AD, why hippocampal sparing AD is more prevalent in males, and limbic predominant AD is more common in females. So we next will integrate with Yo-specific GWAS that our generators are generating it, because we think those genetic modifiers can be different in late onset and young onset AD. And we will explore how different glial states interact with these vulnerable cells, vulnerable neurons. And we are requesting more tissue slides to validate these subtype markers with pathological markers. And we will link transcriptional changes to chromatin accessibility to better understand the gene regulation. I would like to thank our collaborators, Melissa from Mayo and Leah from, she moves to Mayo too, and Felipe, Cyril, Daniel, and my PI Jessica, and all lab members in research lab. My poster is in 11, please come and talk with me. Thank you all, and happy to take questions.
Hi, great talk, very interesting data. I was curious about the estrogen part, and if you've looked beyond estrogen at kind of different hormone profiles with like progesterone markers, or sort of how the combination of things may be changing and associated with some of those factors.
Yeah, we didn't look at other. So we didn't look at other hormone rather than estrogen, and we only look at those estrogen specific receptors, because we know that there's an estrogen therapy that treating female and have a bias effect that in early stage when women get AD. And we have an interest to know maybe some targets in the estrogen signaling have different roles and can be more specific that help to improve the cognitive, because when using the whole estrogen, there are some side effects that can like increase the heart disease.
Great, thanks.
Yeah. Thank you.
Any other questions for Dr. Han? Thank you so much for your presentation.
Thank you.
Hello. Our next presenter is, I'm sorry, give me one moment. Our next presenter is Dr. Ki Hu. Dr. Hu is a project scientist in Dr. David Eisenberg's laboratory and has been here at UCLA for some time now. She had an impactful paper in the last year in Nature, and she's going to tell us about that and some of her ongoing work. Her talk is titled "How Short Peptides Disassemble Tau Fibrils in Alzheimer's Disease." Thank you, Dr.
Thank you. Hello, everyone. I'm Ko-Ho from Professor Darren Asmer Lab, and he's also here today. And then thanks for the Turken Symposium and welcome this Alzheimer LA. So I'm very excited to be here to share with you our recent work is how should peptide disassemble tau fibers in Alzheimer's disease. And also I'm very happy I'm the last speaker because thanks to the previous speaker's introduction, so I didn't have much to talk about in my background. So we know that for the Alzheimer's disease, there is two hallmarkers, this intracellular tau neurofiber tangles and extracellular tau A-beta plaques. And then they are caused by the normal aggregation of the tau and the beta. And then when they purify this aggregate and put it in the electron microscope, you find that they are fibers. And then we call that amyloid fibers because they share this common cross beta structure. And this is not working. So if we put this amino fibers in this X-rays, you will see this cross beta diffraction pattern, which related to this interstrand spacing and the interstrand spacing. It is 4.8 and 10 angstroms. And then the feature of these amino fibers is that they have this seating capacity, because if you have this amino fibers, you can use it as a seat, and then you can recruit other tau or beta monomers and template them to form new fibers. So therefore, in the Alzheimer's disease brain, their A-beta and tau pathology can spread from one brain region to another brain region. And then when the scientists, they compare the symptom, the progression of Alzheimer's disease with this spreading of this A-beta and tau pathology, they found that this cognitive decline correlates more closely with fibers of the tau than the A-beta fibers. So also in the year 2017, the Godet group used this high-tech cryo-EM to identify the atomic structure of the tau fibers in AD. And you can see here that each layer of these fibers, they composed of two tau monomers, and they pair together. And then inside these layers, they assemble and twist it and forms these left-handed twist fibers. And then I want to point out is that this paired helical filament of tau, they are found in all AD patients. That means if you have a drug that can inhibit this tau aggregation or disrupt these tau fibers, they can apply to all the AD patients. And then, so based on now, we know that this tau aggregation mechanism and they're related to the Alzheimer's disease. So we're thinking if we can inhibit this tau aggregation or disaggregate these tau fibers, maybe we can provide a therapy for Alzheimer's disease. So based on that, our lab back to the year 2011, we did a pioneer work and we designed a peptide, dipeptide, and the sequence is TRKW, which can specifically bind on the top of the fibers and inhibit this aggregation. And then when I first joined the lab, I'm thinking that I can conjugate this peptide to nanoparticle. So therefore, I want to develop MRI probes for the early diagnosis of the tau Alzheimer's disease. So to do that, I introduced one actocysteine at the end. And surprisingly, I found that this new peptide, they not only can inhibit this tau aggregation, but also can disaggregate tau fibers. And here we test on the recombinant tau fibers, also the tau fibers extracted from the AD patients. So you can see here that the AD tau inhibited with this previous dipeptide, the fiber is still there. But if you inhibit with this new peptide, most fibers disappear. So this is very interesting, and we want to know why. And then you can notice that the only difference between these two peptides is this cysteine at the end. So the first experiment we do is that we substitute this cysteine with other amino acids and to see how things going. And they found that other dipeptides also have this kind of phenomenon. And then they show variance efficiency. And the varying efficiency of the peptide depending on the types of the 7th residue X. That means if your X is hydrophobic amino acids, this shows the best effect. Later is this polar basic acidic. If your last residue is protein, there is no effect. So then we also use this time course EM to see how these fibers disappear, this disaggregation process. And you can see that compared to the earlier time point, if you inhibit this AD tau with this dipeptide, at 1 hour, after 1 hour, there is something growing on the surface of the AD tau. After 3 hours, we see some new fibers formation. So after 6 hours, the fibers keep growing, but at 24 hours, these new fibers disappear along with these AD tau fibers. And they turn to these amorphous aggregates. So at these 48 hours, the samples are very clean. So then we suspect that these new fibers is dipeptide fibers. And it looks like their fiber formation is very closely to the disaggregation of the AD tau. So to prove that, we design an experiment that we aim methylation of the dipeptide to make it no longer self-aggregate. So you can see that this non-self-aggregating dipeptide truly cannot disrupt AD tau. So this experiment proved that our hypothesis that this amyloid-like property of the dipeptide and is very close, is directly correlated to the disaggregation of AD tau fibers. And then we're curious about this structure of these dipeptide fibers, and we found that the dipeptide fibers are right-handed twisted fibers by themselves. And then we use cryo-EM to solve different kinds of dipeptide fibers. You can see that they have different assemble pattern, but these structures, they have these conserved spherical structures. In this square, you will see that. So in addition to that, we also use cryo-EM to solve the complex structure of AD tau and dipeptide. And you can see that compared to this untreated AD tau, this dipeptide treated AD tau, there is extra density here. And then the shape of it, if you remember, is perfectly matched with the steric zipper structure I just showed in the dipeptide fiber itself. So, but I want to point out that this dipeptide, when it also assemble along the AD tau, but it follows the right-handedness of the AD tau. But in their previous, if they form fibers by themselves, it tends to form these right-handed twisted fibers. So this static cryo-EM just give you a static intermediate state of these 80 tau fibers on their pathway to disaggregation. So then we also use this high-speed AM to visualize this dynamic fragmentation process. So you can see that for the P cells of 80 tau fiber, this disaggregation completed very quickly, only within 10 minutes. But it requires several hours incubation, and this time is for the peptides to assemble along these 80 tau fibers. And you may notice that this fragmentation initially happens in the groove of the fibers. And then if you pay attention to this axis of this fragmentation, you may find that this ends curve consistent with talk-like effect. It looks like you just twist the fibers from left to the right. So then based on put all these pieces information together, and then we proposed our strain relief mechanism, and I also summarized here. So first, it is like when you incubate with dipeptide with 80 tau, the first step is dipeptide bind to the 80 tau. And then thanks to their self-aggregation property, it will assemble along the 80 tau fibers, but it follows the left-handedness of the 80 tau fibers. So, but you also remember that the dipeptide fiber itself, they tend to form these right-handed twisted fibers. And that means the assembled structure here is metastable, and they want to change to the right-handed. So therefore, there is strain here. So to some extent, the strain is enough to break the one layer of this 80 tau and fragmented it. So I want to highlight that we think the complex of the fragmentation product is a complex of the tau and peptide. And we confirm that this disaggregating product is non-sitting species in our biosensor cell line. And that means they are no longer sees this aggregation of the tau fibers. So therefore, it can be a very good drug. You remove these tau fibers, but not generate more Cs. So finally, I want to acknowledge the whole David Eisenberg lab, especially my mentor, Professor David Eisenberg, and all our funding support. And I thank you for your attention, and will take any questions. Thank you.
Well, we have time for a few questions. I'll take privilege and ask the first one. So it looks like the peptide binds into the fibril. Do you have any information about the specificity of that? Is it unique to other protein moieties that might also form fibrillar aggregates?
Yeah, we tested on the upstream clean, and we tested other like functional amino fibers. It didn't have much effect. We also tested on other isoform of the tau, because like different diseases have different conformation of the tau fibers. So we found that they have some effect. They have like effect on other tau pathologies fibers, but not on the other amino fibers like upstream clean, which is reasonable because, you know, when I first designed this dipeptide, it is like designed as a specific inhibitor, means that they have specific binding to the tau fibers. So that's the thing.
Other questions? Dr. Samelson.
So what's like the minimal unit of bound fibril and bound drug? Like when it, so you get strain and they break apart. Is there eventually just disassociation of tau and the drug, or are there stable, basically fragments, stable small fibril fragments or something like that that are drug tau complexes?
Yeah, very good question. And then we think it is like, because based on the EM thing, and then we found that they're kind of like lose their amorphous structure. And then based on the high-speed AM, I'm saying that their disaggregating product is very small, and then you're no longer detected by this high-speed AFM. And they go to this kind of like a soluble in the solution. And then, but their molecular weight is large. It's not the monomers. So that's the thing we think is the complex of the dipeptide and the tau. And then later on, we confirm in our biosensor cell line, they're not sitting species. So that means it's kind of like the dipeptide after they fragmented these fibers, they kind of change their fibrous features and make it no longer a fiber, and then therefore they lose their sibling species.
Great, thank you. Thank
Yeah.
you, Dr. All right, this brings us to the end of today's formal symposium structure. We'll return now to the poster session. And I just want to again thank the Alzheimer's LA and the Sam and Ida Turken Foundation for supporting today's symposium. And particularly, I want to especially call out Beth Devermont, who is our donor for today, and she was very generous and sponsored some light refreshments. So there'll be Dee Dee Reese cookies and Corner Bakery coffee outside in the lobby during the poster sessions, and we're deeply grateful again for her ongoing support.